Lab 7. Using a microscope. Introduction. In almost every type of biological research, the microscope plays a fundamental role. Scientists in each field rely on it to study the fine structures of cells and tissues, things too small to see with the unaided eye. Were it not for the microscope, our understanding of life would be far different from what it is today. In this lab, you will learn how to use a compound light microscope to observe structures too small to see with the unaided eye. In future labs, you will observe microscopic organisms using the techniques you learned in this lab. It is essential that you learn how to use and care for your microscope properly. Your teacher will explain how to use a microscope and give you the names of its parts. The microscope rests on a large, stable base. The image is viewed through the eyepiece. Microscope slides are placed on a flat surface above the base called the stage. The stage is attached to the base on a vertical arm. On the stage are clips that hold the slide in place. In the base is a lamp called the illuminator. This adds additional light to the image for easier viewing. However, sometimes it can add too much light and the image becomes washed out, making details more difficult to see. When this occurs, the diaphragm, a rotating dial just below the stage, can be turned. The diaphragm adjusts the amount of light hitting the slide. A slide can be viewed with either the high or low power objective lens. The microscope is switched between high and low power using the revolving nose piece. Focusing the image is accomplished by using the coarse and fine adjustment knobs on the side of the arm, just above the base. The coarse adjustment knob is the larger of the two knobs. It is used to focus images on low power. If you wish to view an image on high power, focus the object on low power using the coarse adjustment knob. Then switch to high power. The image should still be in focus. If it is not, use the fine adjustment knob. The fine adjustment knob is the smaller of the two. Never use the coarse adjustment knob when the nose piece is turned to high power. Your microscope is expensive and fragile. It is important for you to use it correctly to avoid damaging it and to avoid breaking slides or destroying specimens. Always keep both eyes open when you look into the eyepiece. It is important because it reduces eye strain. If you find this difficult, cover your other eye with your hand. This may feel awkward at first, but it will become easier with practice. Keep the lenses on your microscope clean. Never touch them with your fingers. If the eyepiece or objective lenses get dirty, clean them with a piece of lens paper moistened with alcohol. Wipe the lens in a light circular motion and change the lens paper as it picks up the dirt. Make certain that you leave no streaks on the lens. Note, cleaning the lens with anything other than lens paper or wiping too hard will scratch the lens. The magnification of your microscope is equal to the product of the separate magnifications of the eyepiece and the objective. The magnification of each lens is written on the lens case. If the eyepiece is 10 times and the low power objective is 10 times, then the magnification under low power is 100 times. In equation form, this is written eyepiece magnification times objective magnification equals total magnification. Procedure for this lab. Using your microscope. Number 1. From the classified section of the newspaper, locate a lowercase r. Place the r right side up to the slide. Place a small drop of water on the slide. Place one edge of a cover slip on the slide next to the R. Gently lower the cover slip onto the R. To get rid of bubbles, Raise and lower the cover slip until any trapped air is released. Do not press directly down on the cover slip. If there is not enough water under the cover slip, add water at the edge. Number 2. Put your slide on the microscope stage and clip it in place. Position the slide and adjust the diaphragm to obtain the appropriate light for viewing. 3. Turn the nose piece so that the low power objective is in place. Focus under low power using the coarse focus. You should be able to clearly see the letter R on the slide. Number 4. Draw the letter on low power, showing the appropriate details and following the guidelines for lab drawings. Number 5. Center the letter in the middle of the low power field. To use the high power objective, first center the slide and focus. Then turn the high power objective into place. If your microscope is properly adjusted, the image should be almost in focus. Use only the fine focus on high power. Adjust the position of the slide if necessary and focus to see small irregularities in the R. Note, using the coarse focus with high power will break your slide. Number 6. Draw the letter R under high power. Number 7. To learn how to see the depth of a specimen, obtain 3 pieces of colored thread about 1 centimeter long. Place the cross threads in a drop of water on a clean slide. Add a cover slip. Clip the slide in place on the stage. Focus up and down using low power. 8. Draw the cross threads showing the depth of focus under low power. Rotate the high power objective in place. Observe the thread carefully as you slowly open and close the diaphragm. Microscopic measurement. Number 1. You will now measure the diameters of the low and high power fields. This will enable you to estimate the actual size of the specimens you will observe later. 2. Place the micro ruler slide on the microscope stage so that the millimeter scale is facing up. Locate the 1 millimeter markings, the largest divisions, under low power. Place 1 millimeter marking of your ruler at the far left-hand side of the low power field so that it is barely visible. You should see 1 other millimeter marking in your field of view. This means that your low power field is between 1 and 2 millimeters in diameter. Estimate the fraction of the second millimeter that is in your field of view, 0.5 or 0.6 or 0.7, etc. Add this to 1 millimeter. Now you know the diameter in millimeters of your low power field of view. 3. Make a drawing of the ruler in the field of view on low power. 4. Make sure the ruler is in focus on low power. Rotate the revolving nose piece to high power. Adjust the fine adjustment knob if the image is out of focus. Use the micro ruler to estimate the diameter of the field of view on high power. Number 5. Many of the specimens you will observe under the microscope will be smaller than 1 millimeter in size. Because of this, microscopic measurements are often expressed in microns or micrometers. 1 millimeter equals 1,000 microns. To convert your diameters to microns, multiply them by 1,000. Number 6, make a drawing of the ruler in the field of view on high power. Include size estimations in microns. Microscopic observations. Number 1. In addition to whole cells, you will often wish to observe the parts of a cell. A good example of a part of a plant cell is a grain of starch. The potato stores energy made by photosynthesis are grains of starch. Starch grains appear white in the normal plant cell and are not easy to see. To make them more visible, you will stain them with iodine, which turns them black. Stains are commonly used in microscopy to make certain types of cells or cell parts easier to observe. Number 2, prepare a wet mount of starch grains by cutting a piece of potato. Then, using a single-edged razor blade or a scalpel to gently scrape the surface of the potato, not the skin. You should see a whitish liquid substance on the edge of the blade. You do not need very much, just enough so that you can barely see it. Caution! Be careful not to cut yourself with the razor blade or scalpel, and cut away from yourself. Number 3, place a drop of this fluid onto a clean slide and spread the drop to make a circle about the size of a dime, and add 1 drop of iodine and place a cover slip on top of the iodine. Examine your slide under low power. You will see numerous black circles and ovals. These are starch grains from the potato. Move the slide around to find a field that has about 100 starch grains. Number 4, draw a diagram of what you see under low power. Label the starch grains. Number 5, estimate the average size of a starch grain. First, determine how many starch grains are needed to reach from one side of the visual field to the other. Divide this number into the diameter of your low-power field. This number is the average size of a single starch grain. Number 6, make a wet mount of an organism in pond water. You may need to add some portaslow to the drop before adding the cover slip to slow the organisms down. Number 7, see diagram. Number 8, draw a diagram of the organism under an appropriate power. Estimate the size of the organism and include it on your drawing.